As we discussed last time, mitosis refers to cell division, and mitosis is occurring anywhere in a plant where you have cell division going on, which means basically in the meristems of the plant is where you see this process. Cells that are not actively dividing are said to be in interphase, and a mature cell could said to be in interphase. However, cells that are found in meristems, although they're not necessarily dividing all the time, just prior to dividing and while still in interphase, they go through some preparatory steps so that they can divide properly. And so during interphase, just prior to undergoing mitosis, the cell has to do several things. And they typically increase in size, and it does this by just producing more cytoplasm. It also divides its organelles, or it reproduces its organelles. We'll say makes more organelles, so that, for example, each offspring cell will have an appropriate number of mitochondria and ER and so on and so forth. Probably the most important thing, as we talked about last time, is the DNA must be replicated. In other words, it has to be copied so that each offspring cell ends up with its own complete copy of the so-called owner's manual. So once all three of these things have been accomplished, a cell is now ready to undergo what is called nuclear division, or mitosis. Now, mitosis gets broken into several different phases, and each phase has its own name. And if we start with interphase, although it's not technically part of mitosis, I'm going to go ahead and write it here anyway, though. We start with an I for interphase, and the next one is referred to as prophase, in which certain things happen. And then the next phase, certain things happen, is called metaphase. Then we have one called anaphase. And finally, we have telophase, which wraps everything up, at which point we will then have two complete cells. So to remember the order in which these phases occur, you can just remember this made-up word here, IPMAT, I-P-M-A-T, and I'm sure there's a few mnemonic devices for this. You could probably Google up something and find out some catchy little sentence that you could remember. I've just always remembered IPMAT, and that helps me remember the order. So we've already talked about interphase. Let's talk about the first phase that is truly part of mitosis, and that is the P for prophase. And a lot of these names, I'll give you some little hints on how you can remember what's going on. Pro means pre, and as it says in your notes, this is the longest phase, and this is the phase during which we actually start to see the visual evidence that this cell is about to undergo division. The DNA coils on itself and it thickens, and we actually start to see chromatids forming, or chromosomes, excuse me. The chromosomes form, complete with centromeres. And another thing that happens is the nuclear membrane dissolves. So nuclear membrane disappears. And then finally, as outlined in your notes, something called the spindle fibers begin to form. This is referred to as a spindle apparatus as well. And if I were to draw a cell that this is happening in, it would look something like this. The nuclear membrane would have dissolved away. We'll now have a number of chromosomes. Let's say that this plant has 4 chromosomes in it. The spindle apparatus, or the spindle fibers, they start to form from opposite ends of the cell, and these fibers are just microtubules, and the fibers attach to the centromeres. Now, this spindle apparatus is important in that it is what is going to control the movement of these chromosomes, because one half of each chromosome is going to need to end up on either end of this cell so that the cell can divide down the middle, and each offspring cell will have its full copy of the owner's manual. So that's it. That's prophase. The nucleus dissolves, the chromosomes connect up with the spindle fibers that start to form, and that brings us to the next step, or the next phase, which is called metaphase. Meta means half or middle, and that refers to the fact that our chromosomes that are connected to the spindle fibers get lined up in the middle. I'll draw a few spindle fibers, okay, and they're connected at the centromeres to these spindle fibers, and the spindle fibers lengthen or shorten and basically get them lined up in the middle. So as it says in the notes, the chromosomes align at the cell equator, and then at the end of metaphase, our cell starts to look like this, and we'll see that, as I'm drawing it here, the chromosomes have disconnected at the centromeres. In other words, the sister chromatids are no longer connected by the centromeres. And so that's metaphase. The centromeres, they line up in the middle, and then the centromeres separate. So next on the list is anaphase, and ana means opposite, and that describes what happens next. These halves of chromosomes, or these sister chromatids, get moved to the opposite ends of, ana meaning opposite, of the cell. They get pulled basically to the opposite ends of the cell by the spindle fibers, which are still attached. Whoops, that one should go kind of like that. At the centromeres. Now, keep in mind here, and I'll refer back to it in a few minutes, there are additional spindle fibers that are still traversing this cell along the center here. But we're mainly concerned with what's going on with the chromosomes at this point. And so you could write for anaphase that the chromatids are pulled to opposite ends of the cell. So anaphase divides them, or pulls them to opposite ends, because the next step is going to divide this cell physically in half, and that is called the telophase. Telophase completes the whole process, and so there's a lot of different things going on, and I'm just going to do a sketch here. We've got 1, 2, 3, 4, 1, 2, 3, 4 chromatids, which are exact copies of each other, by the way, on either end of the cell. First thing that happens in telophase is 2 nuclear membranes form around each group of chromosomes. The chromosomes then begin to uncoil, and they go back into that more loosely packed state, and the reason they do that is each of these newly developed cells has to start using the owner's manual, or reading the owner's manual, to put themselves back together and start functioning as an individual cell. And so in order to do that, you've got to open up the books in the chapter, and so they have to uncoil those chromosomes. And so the chromosomes uncoil, and then you can't even see individual chromosomes anymore at this point. Nucleoli reappear, which are just these little dark centers that you often see in a nucleus, and I think we referred to those when we talked about all the various organelles earlier. The spindle fibers, for the most part, have disintegrated. There are a few remaining, and they're important for one of the next steps here, and that is the formation of what's called a cell plate. The short little fibers of the remaining spindle fibers accumulate cell wall material that's being produced and packaged up into small little packages of ER, and more of this stuff accumulates in the center until it starts to become thicker and thicker, and this is what's referred to as the cell plate. And this becomes a new cell wall, and it grows outward until it connects with the original sides of the cell walls. And behind it, new cell membrane gets produced, and basically at this point, we now have two complete cells. Each of these cells has the proper number of chromosomes. There are 4 chromosomes in this one and 4 chromosomes in that one. The original cell had 4 chromosomes in it. Now, this being mitosis, and each chromatid was copied from the original chromatid, the sister chromatids, the DNA makeup of this cell is exactly identical to the DNA makeup of this cell. In other words, they have exactly the same owner's manual. Now, in the next type of cell division that we talk about, we're going to see how it develops not 2 cells, but into 4 cells, each of which has its own unique combination of the original owner's manual. And so they have different editions of the owner's manual, let's say. But in this case, mitosis, we end up with exact copies, or very nearly exact copies, because mistakes get made when we talk about making copies of billions of base pairs. There's always going to be some mistakes made, but for the most part, they are exact copies of each other. And this basically now means we have 2 cells that are now back in interphase. And these cells can now, there's a P in there, these cells can now differentiate and become xylem, phloem, epidermal cells, root hairs, whatever the case may be. Or if they remain in a meristem, each of them is going to go through a short interphase and likely go through this whole process a second time and make even more cells. To quickly summarize, let me back up and look at a couple actual photographs alongside of the drawings here. So we'll start with prophase, where we first start to see chromosomes. And so here's a photo of a cell in prophase. We can see the chromosomes, but they aren't particularly organized. And moving on to metaphase, where they line up in the middle of the cell. Here's a photo of a cell actually in metaphase. We can see they're all lined up at the equator of the cell. We cannot see in this particular photo the spindle fibers, but it's pretty obvious this is metaphase because of the way they're lined up. Moving on to anaphase, this photo depicts a cell in that phase, and you can see that the chromatids have separated and they're being pulled to opposite ends of the cell. In fact, you can actually see that they're bent backwards just like I drew them here. And that is because of the friction of it being pulled through the cytoplasm. The arms bend backwards as they're being pulled to opposite ends. So there's anaphase. And then finally, a few examples of telophase. And by a few examples, I mean a couple of cells here. And this is early telophase, but you can see the chromosomes have started to uncoil. They're no longer as densely packed. They'll soon form nuclear membranes and a cell plate. And finally, this is a cross-section of an onion root meristem, in which we have a bunch of cells in various stages of mitosis, and we can point out a few here. This would be, as you can see, the chromosomes are starting to condense. This would be prophase. We have these two, which are obviously, well, this one still, this is obviously anaphase. They're being pulled to opposite ends. And here's another good shot of anaphase, or possibly early telophase. And these 2 cells appear to have just formed a cell wall between them, which would be late telophase. And you can look around and pick out some of the various stages if you look closely at that photo for yourself. But that's mitosis.